For the objective, first, to determine the basic principles of gram staining for bacteria differentiation. Next, to examine the gram staining result of AQU3814 coli and Staphylococcus to see how their cell wall structures differ. Then, to demonstrate the gram staining method step by step.
Staining, introduced by Kristen Gram in 1883, differentiates between gram-positive and gram-negative microorganisms based on their color rotation. Gram-positive organisms stain blue-purple, while the gram-negative organisms stain pink-red. Stainers mirror display bacterial morphology and arrangement with common morphology being cocci, bacilli, and coccobacilli. The technique and change image clarify in microscope in technique.
For the material, we have inoculation loop, bacteria in agar plate, crystal violet, gram iodine, decolorizer, safranin, Bunsen burner, sterile water, and also microscope slide. Next, for the method, sterilize inoculation loop. After, drop sterile water and then put colony of bacteria. After dry, heat fix bacteria on slide. Add few drop of crystal violet. After 1 minute, rinse the slide. And then add a few drops of gram iodine to cover bacteria. And after 1 minute, rinse the slide again. Next, add a few drops of decolorize and then rinse the slide again. Next, counter stain by adding safranin stain for 1 minute and then rinse the slide again and let slide dry completely, and the sample will observe after it completely dry.
From the gram staining result that our group have obtained, it is indicated that Staphylococcus aureus appear in purple color, which is suggests gram-positive bacteria, while E. coli appear in pink color, which is suggests gram-negative bacteria.
For discussion, the process of separating bacteria species into two groups, which are gram-positive and also gram-negative bacteria, based on the chemical and also physical features of their cell walls, known as gram staining. The content of peptidoglycan is higher in gram-positive bacteria and also lower in gram-negative bacteria. A genus of bacteria called Staphylococcus is identified as a gram-positive bacteria by the purple color. Gram staining also give gram-negative bacteria such as E. coli a pink color. Gram-negative bacteria are known for their absorption of the counter stain safranin, which cause this color. The main distinction is the construction of their cell walls. Gram-positive bacteria have a thick peptidoglycan with no outside lipid membrane, whereas gram-negative bacteria have a thin peptidoglycan layer with an outer lipid membrane.
In conclusion, E. coli has a thin cell wall with less peptidoglycan and an outer lipid membrane, making it resistant to the crystal violet stain and taking up the counter stain safranin, resulting in the pink color. For the Staphylococcus aureus has a thick cell wall with lots of peptidoglycan and no outer lipid membrane, allowing it to retain the crystal violet stain and appear purple.